and I am going to elaborate on the topic strain improvement of industrially important microorganisms. So what is meant by strain improvement? Strain improvement is a process in which we are going to alter the microorganism's genetic makeup so that it gives higher yield of the desired product. So for it is very necessary for us to change its genetic makeup. Only by changing its pH, media formulation, temperature, aeration, agitation, such parameters, it is not going to change into it is not going to give higher yields. So for that, there are certain processes. So how is this strain improvement done? And what is the purpose behind it? What is the purpose? Strain improvement, it gives higher yields of the desired product and it makes the process more economical. So, there are some techniques by which we can improve a certain strain, which are first, mutation, second, strain improvement by genetic recombination or by gene transfer, and third is strain improvement by genetic engineering. So let us start with the first, strain improvement by mutation. So what is meant by mutation? Mutation is a sudden hereditary change in the genetic makeup of a certain microorganism or any individual. So what we can do is we can use certain mutagenic agents such as UV radiations, alkylating agents, nitrous acid base analogues, ionizing radiations to change the genetic makeup of a certain bacteria. So for that, we can say that if I have a microorganism that I have isolated from the wild and it gives a certain yield, which is somewhat 1.75 grams per liter. Now, if I expose the same bacteria to mutagenic agent like UV radiations, these UV radiations might change the genetic makeup of that bacteria, and which will lead to two types of bacteria. One which will give enhanced product synthesis, and the other type which can give decreased product synthesis. So the wild type, which will be changed into the mutant type, and then a strain is selected which will give enhanced product, which is this one. For this, selection is very tedious. This process is very tedious, but some microorganisms survive lethal dose of such mutagenic agents, and some microorganisms cannot survive lethal doses of such mutagenic agents. Those which survive lethal doses of mutagenic agents, they mutate, they transform into a mutant microorganisms, whereas organisms which do not survive these high levels of lethal doses, they die in that case. So it is a very complicated job, but again, for the selection of the mutant which is giving us higher yields of the product is again a tedious technique. These mutations can lead into first major mutation, where in major mutation profound changes takes place in the microorganism, that is changes in the morphological and biochemical properties takes place. This is called as major mutation. A major mutation in which profound changes, that is everything changes in the microorganisms, morphological and biochemical properties. And the second one is minor mutation. There is only slight change in the biochemical characteristic of that microorganism. This is called as major mutation and minor mutation. So for example, if we take an organism, a fungus, Streptomyces species, which is wild, and expose it, and it produces antimycin A, which is an antibiotic. If we expose the same fungus to a certain mutagenic agent, and this Streptomyces species will show absence of pigment, alteration in dense collineation, which is an example of major mutation, and it will give yield of the same antibiotic 0.75 to 9.75 grams per liter of the substrate. So how are these improved strains selected. For the selection of auxotrophic mutant, it is comparatively easier than selection of non-auxotrophic mutant. So for the selection of auxotrophic mutant, there is a technique called as penicillin technique, which we are going to see. Penicillin technique it is used for the selection of auxotrophic mutant. In this, what we can do is, first, take a flask consisting of minimal medium, which consists of, in which we can inoculate both the non-auxotrophic, that is wild bacteria, as well as or fungus, or and both auxotrophic and non-auxotrophic organisms. We add non-auxotrophic and auxotrophic mutant. Now we don't know if if there is auxotrophic mutant in the minimal medium. So what we are going to do is, we are going to add penicillin antibiotic in the same flask and let it grow. We are going to incubate it. After incubation, what will happen is, the flask is containing both non-auxotrophic and as well as auxotrophic mutants, but only non-auxotrophic mutant will grow because penicillin acts on growing cells. It acts on growing cells. So the non-auxotrophic mutant, as they divide, they will get immediately killed, because as we know that penicillin it acts on the cell wall synthesis of the bacteria, and it will eventually die, the non-auxotrophic mutants. Whereas the auxotrophic mutants will stay in the dormant state. They will not divide, they will not grow. They will just be in that medium. Second technique, now we can select, okay, now we can use the same broth as an inoculum for auxotrophic mutant. This we can select. This is first technique. Second technique we can use is, take a suspension which consists of the selected organism and add it in complete media. Take the same suspension of A, which is auxotrophic mutant, and add it in complete medium. Now complete medium has growth factors which are required by auxotrophic mutants. So now auxotrophic mutants will grow in the medium, complete medium. Now what we can do is, we can centrifuge the cells, centrifuge the broth, separate it, wash it, and place it in a broth containing, penicillin. incubation, and after incubation what we can do is wild cells will be killed here, and for the selection of the auxotrophic mutant we can use replica plate technique. In which we make plates of minimal media and complete media. For replica plate technique, what we do is we take the suspension and spread 0.1 ml of the sample on the plates, on the complete medium plates, and after spreading what we do is we incubate it. So after incubation at a certain temperature and time period, what we will find is growth of certain organisms, certain colonies on the plate. For example, we have 4 different colonies on the complete medium plate. Now what we can do is prepare a minimal media which lacks a growth factor which is required for the auxotroph, and now what we can do is replicate it by using a replica block and velvet cloth. What we can do is simply take an impression of the complete medium onto the minimal medium, and then after incubation what we might find is there is growth of certain colonies and there is absence of growth of certain colonies. From this we can conclude that these two colonies are auxotrophic mutants. Why? Because they don't grow on minimal medium. Auxotrophic mutants are such organisms which cannot synthesize their own growth factors, and hence they have to rely on external growth factors, and hence they don't grow on minimal media. So this complete media can, by comparing the both of the plates, we can say that this is the auxotrophic mutant, and we can select it and store it, subculture it and maintain it, and then use it industrially. Then second technique is genetic recombination. This is strain improvement by mutation. Now we are going to see strain improvement by genetic recombination. Strain improvement by genetic recombination, which is B. Where what we can do is gene transfer can be carried out by conjugation, transformation or transduction. For that, first we can use it. For example, if we use Streptomyces species 1, which produces antibiotic 1 or XYZ, and there is another Streptomyces species which produces antibiotic 2. Okay, what we can do is combine these both, form a hybrid and produce another fungus which produces Streptomyces antibiotic 3. That is the combination of one gene and second gene, which will lead to the formation of third gene, which might give other new characteristics. For this, can use techniques like processes like for genetic recombination we can use processes like transformation, transduction and conjugation. Firstly we are going to see genetic recombination by conjugation. In conjugation, when two bacteria containing, we suppose this is a donor cell and this is a recipient cell. If a bacteria consists of a plasmid that has an extra gene which gives it an ability to produce a certain antibiotic, then with the help of ciliated appendages like pili, both these bacteria come in close contact with each other and the chromosome is transferred in the recipient cell. This is R cell, this is D. D stands for donor, R stands for recipient. Now, what happens is bacteria has its own chromosome, and with the chromosome, the transferred DNA, the transferred chromosome, some part of it might be cleaved by the enzymes and some part of it might form a crossover with the chromosome that is present in the bacteria, which will be introduced in the bacterial chromosome. This cell is called as recombinant cell or recombinant DNA. Second is genetic recombination by transformation. Similar type of mechanism takes place, but here shredded DNA or pieces of DNA are introduced into the cell by increasing the porosity of the bacterial cell, and then this naked DNA forms a crossover with the existing bacterial chromosome, and it is introduced into the chromosome. Now this chromosome will replicate, and the additional characteristic, if it is an antibiotic producing gene, then this chromosome will be responsible for that extra additional characteristic to that cell. Third is genetic recombination by transduction. For transduction, there is involvement of bacteriophages. Bacteriophages are those organisms which infect a bacterial cell. Bacteriophages. Third is by transduction. In transduction, virus, what happens is a bacteria example, a virus, a bacteriophage which has this blue DNA, what happens is it is going to attach onto the surface of the bacterial cell, and for its replication it is going to insert its genetic material inside the host cell. Now what happens is this DNA or RNA, it gets DNA, it introduces or incorporates itself into the bacterial chromosome, and now it replicates. While replicating, now this bacteria has bacteriophages DNA, and now it is going to replicate. When it replicates, it is going to form all the proteins that are present in the viral capsid. So it is going to produce virus particles into the host cell and pack its own genetic material along with it. Now, if the bacteriophage undergoes lytic cycle, this cell will burst, and all these viral particles will release into the environment. Now these viral particles with some part of the host gene, now this will go and attach onto different cells which are present in close proximity of the same bacteria, and now this cell is going to attach onto a new cell, and it is going to transfer its genetic material, which consists of both, into the bacterial cell. Now this is going to incorporate itself into the bacterial cell, and this will replicate, forming a crossover and then finally forming a recombinant DNA. So these are three processes of genetic recombination, which is transformation, transduction and conjugation. And the last one, that the technique that is used for strain improvement is strain improvement by genetic engineering. Here what we do is we use different tools required for the transfer or introduction of gene into the host. For that, for example, insulin producing gene, which is present in a human cell. This is example, this is DNA, and this is a human cell. Okay. Now, there is another cell, bacterial cell, which consists of its own chromosome. So this is E. coli. It has plasmid DNA as well as chromosome, its own chromosome. So, with the help of certain restriction enzymes, which help in the cleaving of the cutting of this plasmid DNA, what we can do is form a nick in the plasmid DNA, and from a human cell, the DNA, from that DNA, what we can do is gene responsible for producing insulin is cut and used. Now, both of these are, after cutting it with the restriction enzymes, with the help of ligases, we can form a DNA molecule, plasmid DNA, which has insulin producing ability. Human insulin producing ability. Now, what we can do is transfer it into a vector which will help in the cloning of the same plasmid DNA. Cloning it as in it is going to, if the vector, if the plasmid DNA is self-replicating, it will self-replicate into many copies, and then it will transfer into, it is transferred into another bacteria and this bacteria, suppose the same bacteria, E. coli, what we are going to do is, after reintroduction, we are going to grow it into a medium which will help in the production of these cells. Now, this broth might contain insulin, is what we can interpret. And there are certain techniques for downstreaming which will help in the purification of insulin, and finally the product is packaged and manufactured, packaged and then sold in the form of dried powder. Disadvantages are the foreign DNA may be unstable and it might get eliminated by the restriction enzymes present in the bacterial cell, and there are no cloning vectors specifically available for industrially important microorganisms. So these are two disadvantages of genetic engineering, but this is the technique by which we can, this is genetic engineering. And so we have covered strain improvement by mutation, genetic recombination, and by genetic engineering.